Dear students, this is 11th Biozoology video class, Chapter 9, Part 2. Chapter 9, Part 2, Locomotion and Movement. Last video class, we have seen types of movements, and also we have seen structure of skeletal muscle. We have seen structure of skeletal muscle. We have seen here. This is the structure of skeletal muscles. The skeletal muscles, each muscle fiber is thin and elongated one. So it is most of them taper at both end, one end or both end. Muscle fiber has multiple oval nuclei just beneath its plasma membrane or sarcolemma. The cytoplasm of muscle fiber is called as sarcoplasm. Just underline one word. Cytoplasm of muscle fiber is called as sarcoplasm. So that sarcoplasm contains glycosomes, myoglobin, and sarcoplasmic reticulum. We'll see one by one. What is myoglobin? It is a red colored respiratory pigment of muscle fiber. It is similar to hemoglobin and contains iron group that has affinity towards oxygen. So myoglobin is the red in color. So they have respiratory pigment in their muscles, and they similar to hemoglobin as we have in our RBC cells. That hemoglobins are just they attract towards the oxygen and serve as the reservoir of oxygen. Reservoir means storage of oxygen. The next one is the one glycosomes. So the glycosomes are the granules of stored glycogen that provide glucose during the period of muscle fiber activity. Actin and myosin are the muscle proteins present in the muscle fiber. The next we are going to see structure of sarcomere. We're going to see structure of sarcomere. Here, this is the organizational level of skeletal muscles. Structure of sarcomere. Here, this is the muscle. The muscles, they have the basic structural functional unit of muscle we call as a sarcomere. This I'll explain to you what is sarcomere, what is myofibril. Just we'll see all these things. Along the length of each myofibril, there are a repeat series of dark and light bands. There are repeat series of dark and light band. Here, this is we call as a dark band and light band. Dark band and light band. Can you all see dark band and light band? The dark band is called as a A band. That is an isotropic bands. And light band we call as a I band. That is isotropic bands. They'll ask you one word. A band is the one and isotropic bands. Whereas I band, that is a light band, we call as a isotropic bands. They are perfectly aligned with one another. They are perfectly aligned with one another. This type of arrangement gives the cells a striated appearance. Now we'll see the dark band. What is dark band? We'll see each dark band has a lighter regions In the middle we call as H zones. The middle we call as a H zones here. So this is we call as a dark band and light band. So this is the structure of myofibril, I band, A band, I band. The middle of the A band there is less density we can say, that is we call as a H zones. So here the dark band has a lighter regions called as a H zones. Each H zones is bisected vertically by a dark line called as a M line. The light I bands also have a darker midline area called as a Z discs. The Z discs means Swiss chasuble. Swiss Chasensbild. The scientist name, this German scientist name. He only found out this one. That's why it's also Z discs. The myofibril contains a contractile elements. Myofibril contains contractile elements. The sarcomere, which is the functional unit of the skeletal muscles, already told you, sarcomere is the one structural unit of the skeletal muscles. The sarcomere is the regions of myofibril between two successive Z discs. It contains an A band with half I band at each end. Inside the sarcomere, two types of filaments are present, namely thick and thin filaments. So that's all about the structure of the sarcomere. Here we'll see the thick filaments extends the entire length of A band. The thin filaments extend across the I band and partially into the A band. The invaginations of the sarcolemma forms a transfer tubules and they penetrate into the junctions between A and I band. A and I band. So this is what the structure of skeletal of sarcomere, a myofibril we can say. The next one is the one here. Can you find this tabular column? Can you mark it at one word they'll ask you? The muscle terminology, the cell, plasma membrane. Sarcolemma we call as a plasma membrane. The cytoplasm we call as a sarcoplasm. The endoplasmic reticulum we call as a sarcoplasmic reticulum. Important 2 marks, just mark it out. The next we'll see the structure of contractile proteins. Structure of contractile proteins. Okay, here the structure of contractile proteins. The contractions of muscle depends on the presence of contractile proteins such as actin and myosin. Underline actin and myosin in the myofilaments. The thick filaments are again we'll see the structure of contractile proteins. So the proteins which are present in the muscles, that structure we are going to see, that is we call as a structure of contractile proteins. So contractile means the muscles which is going to contract or wrinkle, that is we call as a contractile. So during the positions which undergo for the wrinkle positions, when the muscles undergoing for the contractile, during that time what type of proteins, what are the proteins are used, what is the structure of that proteins we are going to see in this topic. The contractions of muscle depends on the presence of contractile proteins. Such proteins we call as actin and myosin in the myofilaments. The thick filaments are composed of the protein myosin, which are bundled together, whose ends produced at opposite ends of the filaments. Each myosin molecule is made up of monomer called as a meromyosin. Underline, check out your textbook, just underline one word. Each myosin, that's okay, each myosin molecules, that's a type of proteins. That each myosin molecule is made up of monomer. Monomer means single proteins, single type of proteins joined together we call as a monomer, called as a meromyosins. The meromyosin has 2 regions, a globular head with a short arm and tail. The short arm constitutes a heavy meromyosin. I'll show you what is heavy neuromyosin. HMM means heavy neuromyosin. Just underline that one. I'll show you the diagram here. This is the structure of myosin, the proteins which I told you, the contractile proteins, myosin. So this is the structure of contractile proteins of myosin. It's a thick filaments. You can find. So this is the single structure of myosin proteins here. Actin binding sites, heads, ATP binding sites, HMM. HM means what? Heavy neuromyosin. And tail portions form LMM. That's a light neuromyosin. Just underline. The head bears an actin binding sites. Here, this is also actin binding sites. Can you all see actin binding site? And ATP binding site, actin binding site and the ATP binding sites. It also contains ATPase enzymes. So that enzyme splits ATP to generate energy for contractions of muscle. Contractions of muscles. So the thin filaments are composed of two intertwined actin molecules. Okay, the next one is the one actin. The actin has a polypeptide subunit called as a globular here. This is because of actin. This is the structure of actin 1. Actin has a polypeptide subunit called as a globular actin or G-actin we say. The filamentous form are F-actin. Each thin filament is made up of two F-actin helically wound to each other. Here, this is because of two filament actins. Spirally a coiled one we can say. Spirally a coiled one we can say. And spirally coiled we can say. That's we say as a helically wound to each other. Each F-actin is a polymer of monomeric G-actins. G-actins 1. It also contains a binding site for myosin. It also contains binding site of myosin. Here, this is a reed structure of actin. This actin only they have a binding site of myosin. The thin filaments also contain several regulatory proteins like tropomyosin, troponin, which helps in regulating the contractions of muscle proteins with the actin and myosins. So this is the structure of contractile proteins. So there is a one word, important one word, myology. Myology means study about muscles, we call it a myology, we can say. Study about muscles, we call it a myology. The next topic is the one, mechanism of muscle contractions. Mechanisms of muscle contractions. Here, sliding filament theory, you can say. Sliding filament theory. In 1954, Andrew F. Axley and Rolf Niederjerk proposed the sliding filament theory to explain the muscle contractions. So they'll ask you one word, underline, take out your textbook, underline this one. According to this theory, overlapping actin and myosin filaments of fixed length slide past one another in an energy requiring processes, resulting in muscles contractions. The contractions of muscle fibers is a remarkable process that helps in creating force to move to resist a load. The force which is created by the contracting muscles called as a muscle tensions. Once if this myosin actin and just contacting each other means there will be muscle tensions will undergo. The load is a weight or force that opposes the contractions of muscles. The contractions is creations of tensions in the muscles, which is an active process. And relaxation is the release of tensions created by contractions. When the actin and myosin just contact each other, that is because of tensions. Okay, that is because of tensions, we can say muscle tensions. When the release, we call as a relaxations of tensions. The muscle contraction is initiated by a nerve impulse sent by the CNS, central nervous system, through the motor neurons. The junction between the motor neurons and sarcolemma of the muscle fibers called as a neuromuscular junction. Just underline. The junction between the muscle fibers or so neurons because of neuromuscular junctions, a motor end plate. When the nerve impulse reaches the neuromuscular junctions, so there is a proteins we call as acetylcholine. Proteins released. It initiates the opening of multiple gated channels in the sarcolemma. The action potential travels through the T tubules, triggers the release of calcium ions from the sarcoplasmic reticulum. The released calcium ions bind troponin on the thin filaments. The tropomyosin uncovers the myosin's binding sites on the thin filaments. Now the active sites are exposed to the head of myosins to form cross bridge. During cross bridge, the formations of actin myosin forms a protein complex called as acetomyosin. The utilizing energy released from the hydrolysis of ATP, the myosin head rotates until it forms 90-degree angles with a long axis to the filaments in these positions of myosin. Here you can find, so the top one we can say as a actin one, the bottom one, this is because of myosin. When our muscles need energy during that time, this myosin head they contact with the actin. During that time, the energy have been released, the ATP, energy have been released from the ATP molecules. And because of that, the muscles undergoing for the contractions and get due to getting energy from this one. And while they contact each other, this myosin, they bend up to 90-degree angles with the long axis of the filaments. In this position, myosin bind to an actin and activate the contractions relaxation cycle, which is followed by power stroke. So this is the structure of the mechanism of muscle contractions. You can go through. So go through this diagram where you can understand. If you have any doubt, you can contact me. The next topic is the one, the schematic presentations of muscle contractions. Whatever we are so far seen, just that's they given as a arrow mark, point wise, just you can learn it out. Then types of skeletal muscles contractions. So there are two primary types of muscle contractions. Other one is the isotonic contractions, another one is the isometric contractions. What is isotonic contractions? Iso means same, tone means weight or resistance. In isotonic contraction, the length of the muscle changes but the tension remains constant. Mark it 2 marks, important. Here the force produced is unchanged. Example, lifting dumbbells and weight lifting. Then isometric contractions. Isometric contractions, the length of the muscle does not change but tension of the muscle changes. Here the force produced is changed. Example, pushing against a wall, holding a heavy bag. The types of skeletal muscle fibers. Here oxidative fibers, then red muscle fibers, glycolytic fibers, white muscle fibers. So these are the skeletal muscle fibers. The muscle fibers can be classified on the basis of their rate of shortening, either fast or slow. The way in which they produce the ATP needed for contractions, either oxidative or glycolytic. The fiber containing myosin with high ATPase activity are classified as a fast fibers with a lower ATPase activity are classified as slow fibers. The fibers that contain numerous mitochondria and have high capacity for oxidative phosphorylations are called as the oxidative fibers. Such fibers depends on the blood flow to deliver oxygen and nutrients to the muscles. The oxidative fibers are termed as red muscle fibers. Mark it 2 marks. The fibers that contains few mitochondria but possesses a high concentration of glycolytic enzymes and are large stores of glycogens are called as glycolytic fibers. The lack of myoglobin gives a pale color to the fiber, so they are termed as a white muscle fibers. So slow oxidative fibers, just you can go through underline, and fast oxidative fibers you can underline, and fast glycolytic fiber, just underline this one. Then next topic is the one remaining wall. I think remaining wall, the types of joint. Next topic is the one types of joints. Types of joints, take out that one, types of joints. Next topic is the one types of joints. Joints are essential for all types of movements performed by the bony parts of the body. The joints are points of contact or between bones. Contact between bones. Sometimes they are playing a protective role in the process. Force generated by the muscles are used to carry out the movement through the joints, which helps in the human functional activity of daily living and ambulations. The joints acts as a fulcrum of lever. The fibrous joints, they are immovable fixed joints which no movements between bones is possible. The suture of flat skull bones are fibrous joints. Suture means fixing plays a hollow-like structure, that is because of suture of the flat skull bones are fibrous joints. The next joint is the one cartilaginous joints. They are slightly movable joints in which joint surface are separated by cartilage, and slightly movements is only possible. The next one, synovial joints. They are freely movable joints with articulating the bones are separated by cavity which is filled with the synovial fluids. Just go through the different types of joints. So with this we have just completed chapter 9. Remaining topics all omitted, one reduced topic, just go through. So with this video we have completed chapter 8. With this video we have completed chapter 8. If you have doubt, we can discuss in the direct class. Go through this topics, just learn and write this one, this schematic representations of muscle contractions. Learn and write this one, important 5 marks. If you have any doubt, we can have direct class clarifications. Okay, bye, take care.